Friends, today I will be discussing about the sleep regulation by the hypothalamus, because this is one of the topic which has a wide diversity involvement of a number of structures in the brain, the encephalon, the medulla and the reticular formation. As you see here in this cartoon, the various things which regulate the sleep and its connectivity to the hypothalamus. I will look into this in a comprehensive manner in this discussion. As we have seen, because I have been discussing about hypothalamus and I have covered the parts and functions of hypothalamus in one of my lectures, followed by the hypothalamic regulation of endocrine functions and hypothalamic regulation of temperature and the hypothalamus in the feeding and satiety that I have covered in one lecture, and I have covered in one lecture the regulation of water intake and thirst mechanisms by the hypothalamus. Today I will be looking into the hypothalamus in sleep regulation. In subsequent classes, these are the topics I have in my mind, which I will be covering in the coming lecture series. That is the regulation of the circadian rhythm, the hypothalamus as a component of the limbic system, and autonomic regulation by the hypothalamus. Now, let us consider the topic hypothalamus in sleep regulation. This topic I want to address by answering the following 10 questions. These questions are: what is sleep? What are the brain areas of sleep? What are the neurochemicals involved in sleep and wakeful state? Why we are awake? How sleep is induced? How sleep wakefulness cycle is maintained? What are the NREM, non-rapid eye movement sleep centers? What are REM sleep centers? How NREM to REM transition is brought about, and what mechanisms enter the sleep cycle. So this entirely covers all the areas which I am planning to cover today. Now we start with the question number 1. What is sleep? Sleep is a behavioral state represented by a temporary loss of consciousness that differs from a coma or deep anesthesia because it is promptly and completely reversed. I repeat again, sleep is a behavioral state represented by a temporary loss of consciousness that differs from a coma and deep anesthesia due to its prompt and complete reversibility. This does not complete the complete definition of the sleep. Now, if we consider the sleep, sleep is not a passive phenomenon, or it is not a homogeneous event because of the reduced activity of certain parts of the central nervous system. If you consider the sleep, it is the odd mosaic or a complex mixture of physiological events happening at different levels of the neuroaxis involving the central and peripheral nervous system over a period of time. That's what the total sleep is about. Now, we move to the next question. What are the brain areas of the sleep? Now, I have made here, we have three areas of sleep which we have a lot of researches and the documentation for this. The one is the reticular formation. The second is the hypothalamus. Third is the thalamus and cortical projections or thalamocortical projections. Now consider the reticular formation. In the reticular formation, we have a number of nuclei. The midline Raphe nucleus, which is serotonergic, which ascends up and projects to the hypothalamus and thalamus. Locus ceruleus, which is a norepinenergic and projects to the hypothalamus as well as to the thalamus. The mesolimbic tegmental area, this is dopaminergic area. And we have the ventral periaqueductal area, this is also a dopaminergic area. Now we have another sleep center with a cholinergic transmission with acetylcholine as a transmitter. This is from the pontine and mesenchymal area. So these pontine and mesenchymal area include lateral dorsal nucleus and pontopeduncular nucleus. So these first four, Raphe, locus ceruleus, mesolimbic, ventral periaqueductal area, even I include here tuberomammillary projections. And all these things are put together as a aminergic components of the sleep. Now we have another component. This is a cholinergic component which is present in the pontomesenchymal region. Now, in case of hypothalamus, we can primarily identify three areas. One, the lateral hypothalamus and some part of the posterior hypothalamus, which have orexinergic and melanocyte concentrating hormone or MCergic neurons. And by the way, the lateral hypothalamus is concerned with the feeding. You can just see that orexinergic are projecting onto the feeding center if you are looking at the functions of the lateral hypothalamus. Then we have another important area that is in the ventral. This is the posterior hypothalamus, the ventral hypothalamus. We have another area, the ventrolateral preoptic area. Now you can see this. This is the ventrolateral preoptic area. These are the posterior and the lateral hypothalamic nucleus. So this is here in the posterior region, and this is here in the anterior region. And these ventrolateral preoptic area known as VLPO, this has many projections, and this receives the inputs from adenosine, melatonin, Gaba, and also receives the inputs from prostaglandins, leukotrienes, cytokines, and a number of other neurochemicals. Then we have another area in the hypothalamus which is supracosmetic nucleus. This is concerned with the maintenance of the biological clock. That means our whatever the duration of the day is fixed in the supracosmetic nucleus in this day. So whatever we have to do, so those are all triggered here. This regulates the circadian rhythm. So this may be ranging from 23.5 to 26.5 or 24.5 hours a day. Little bit variation. Then we have a third area that is from the thalamus. This thalamic area includes the reticular nucleus of the thalamus, interlaminar nucleus of the thalamus, and other thalamic nuclei which are concerned with the limbic components, and those are all are there. They are projecting onto the all the areas of the cortical areas of the brain. They are glutamatergic. Now, what is the, because the question was what are the various areas of the sleep. Now we consider the areas of the sleep, the reticular formation or reticular areas. So now in this reticular area I have already listed the locus coeruleus, that is norepinephrine. This is raphenucleus, the serotonergic, and the tuberomammillary nucleus histaminergic, and we have these the ventral periaqueductal gray, that is dopaminergic, and these two pontopeduncular tegmentum and lateral dorsal tegmentum, these are cholinergic neurons. Now we have a perifornical area, that is the orexinergic, that is projecting onto the lateral hypothalamus. So these are all the areas, they are activated or they give the projections to the entire cortex, and definitely those projections goes via the thalamus, and they will spread around the entire cortical mantle, and they will keep this state activated. So the key elements of the ascending arousal system are diffuse excitatory projections to the cortex as shown here. Now one point is that the decreased impulse traffic going on this reduces the alertness or arousal. That's one point. So that means the alertness response or arousal response in our body is because of the active participation of these areas to keep us awake or alert. Now, what are the neurochemicals involved in sleep and wakeful state? Now I have listed major neurochemicals here. So now first one is the glutamate. This glutamate is a neurotransmitter from the sensory collaterals, thalamo-peduncular projections, the cortico-reticular projections, and the projections from the lateral parabrachial nucleus coming from the reticular formation. Now we have norepinephrine coming from the locus coeruleus, serotonergic coming from the raphenuclei, and histamine coming from the tuberomammillary nucleus, dopamine coming from the ventral periaqueductal gray, acetylcholine coming from the pedunculopontine and lateral dorsal tegmentum area, orexin from the perifornical area, MCH, this I have not shown, the melanocyte concentrating hormone, which is in the lateral hypothalamus and in the posterior hypothalamus. GABA, it is distributed extensively between the various nuclei. So primarily on the VLPO, that is ventrolateral preoptic area, lateral hypothalamic area, posterior hypothalamic area, supracosmetic nucleus, and they are reciprocally interrelated, and with the reticular formation. The melatonin coming from the pineal gland to the supracosmetic nucleus as well as to the VLPO, and even to an extent with the lateral hypothalamic area. The galanin, which is the galanin is coming from the VLPO area, and this is again now it has been said that it is a REM triggering chemical. So now REM triggering chemicals, not only this, MCH is also one of the candidates there. Then we have a adenosine molecule, and this is coming from the perifornical area. This is also this is somonogenic. Then we have other chemicals like nitric oxide, cytokines, the prostaglandins. They all accumulate around surrounding the VLPO, and they all produce a somnogenic. Uh, just to recall the fact that the cytokines and prostaglandins are elevated in the infectious state or infection or any of those activities. So that will, whenever we are running fever or we are having fever, we feel sleepy. So you can attribute this to this chemical. Now coming back, what are the various the problems associated with the neurochemicals? Problems in the sense of what is the manifestations with the neurochemicals? So just to revise or remind, the one is the narcolepsy. Narcolepsy is an irresistible desire to sleep. It is a hypothalamic disorder, and it results from the selective loss of orexin releasing neurons or whatever the orexin producing neurons in the forebrain, and that accounts for excessive daytime sleepiness and fragmented sleep. So that means orexinergic neuron loss in the forebrain area. Then the second condition, cataplexy. Cataplexy is a disorder associated with muscle weakness or a loss of muscle tone or atonia. Sudden muscle weakness without loss of consciousness. That is cataplexy. Is associated with this disorder, and that may be coming from the cholinergic neurons present in the pons nucleus, which is inhibiting the muscle tone. So when we talk about the REM sleep, you will try to get the idea about that. Then we have antihistamines, because histaminergic blockers, they will produce a drowsiness and sleepiness. So we all have sometime or the other have experienced the drowsiness after taking antihistamine for some other treatment. The melatonin is a drug, or it's a chemical secreted by the pineal gland. It's the hormone secreted in the dark. So it is in the, as we experience with the jet lag, or it is known as a somnogenic, or it induces the sleep. So melatonin induces the sleep. Maybe I will cover with that. Then serotonin is a complex structure. One, it has one activity which is excitatory, which gives the alertness, and some the serotonergic receptors, they are somnogenic. So that means it produces the sleep. So that means it may be because it acts as a substrate for the melatonin synthesis. That is how it becomes somnogenic. Or it may be activating through the other 5-HT receptors, which are producing the sleep. Now we have the GABA. It is the major sleep inducing neurotransmitter, and if you are looking at any of those GABA agonists, they will produce the sleep, like barbiturates. Barbiturates is a agonist at a barbiturate binding site on the GABA receptor complex. Now the GABA receptor complex has a three components. One is a muscimol binding site, that is a agonist binding site, and the other is a barbiturate binding site, that barbiturates bind and open the channel and opens up the chloride. And we have a diazepam or a benzodiazepine binding site, where the diazepams act. So that means we have a barbiturates and diazepam as a agonist for a GABA that will induce sleep. Now, I have already mentioned sleepy state. We during the fever and infections, wherein the cytokines, thermal stimulation, the prostaglandin accumulation, and all those things increase the sleep. Other things, what I have not mentioned here is adenosine. Adenosine is a molecule when it is accumulated during the entire day's work because of the increased metabolism. That is one. Or the other thing is the activation of the feeding center, maybe by the orexins, that would also stimulate sleep. So that means adenosine stimulate. That means a large amount of adenosine accumulation increases or induce the sleep. Okay, so as I go further, you will try to know about these chemicals in a detailed or in a manner in which how they are interacting with the sleep. Now, why we are awake? So that is the very fundamental question when we are talking about the sleep. Now, we are awake because already I have seen these are the arousal centers in the reticular formation. These arousal centers are the locus coeruleus, the the raphe the periaqueductal gray, and tubero-mammillary area, and all those things are active. They are active, and they will activate the entire cortical, cerebral cortex, the entire cortical areas. And these activation through the cerebral cortical areas that would produce the arousal state. Now, so these areas, if you are looking at these yellow arrows, if you are looking at the yellow arrows, so they will go through the thalamus, and again these thalamus they will project onto the entire cortical mantle. That means they reinforce our alertness. So that means they see that the arousal centers are activating us, and we keeps us alert. So now that means the specific areas in the brainstem. These are the areas, the locus coeruleus, what is called a reticular activating system, the locus coeruleus, raphe and periaqueductal gray, and even the ponto or mesenchymal pontopedunculaar centers, and the some part in the tuberomammillary areas. They are stimulated and they are activating the entire cerebral cortex. So this brainstem and hypothalamus are involved. And by keeping the cortex active, that means that keeps us conscious. Now, consciousness is required to perform various functions of our the mind and the body. So that means functions such as memory, speech, cognition, the locomotion, and the looking for food, and many more. The meeting responses, and all those things are performed. So these are the conscious state that brings about the uh, brought about by these reticular formation and hypothalamic areas. Now, so now this is the second part. The orexins also, because basically in the animal kingdom, when the animal gets up, it is the food that looks for the food or the hunting. So looking for the food, this thing, and that is why the orexin neurons. It is thought that the orexins neurons or orexin is going to stimulate these areas in the reticular formation and the hypothalamus. You can just see them. These are the reticular activating system. We have these the locus coeruleus and the raphe and the periaqueductal gray and lateral hypothalamus and tuberomammillary area, and these are the cholinergic components. They are all stimulated by the orexinergic neurons. So the neurons that produces transmitter orexin, they are situated. This is orexin. They are situated in the lateral hypothalamus. Lateral hypothalamus, and this is also known as hypocretin. Orexin is also known as hypocretin, and they stimulate these all arousal centers, and these arousal centers will activate the entire cortical areas. These neurons keep the arousal centers active, helping to stabilize. So that means these orexin neurons further added to the activation of the reticular activating system. What we have seen here, this activating of these arousal centers, further orexin also trying to activate those centers, and to keep us or reinforcing, keep us awake, keep us awake. Now, so now it is not only the these two components. We are awake by the complex set of activities coming from the brainstem, hypothalamus, and the diencephalic structures that would produce us awake. Let us, in this cartoon, I have shown the first thing is I have shown here is the reticular formation. I have included in the reticular formation all those areas, that is the I have listed here parabrachial, the ascending parabrachial nucleus, the roughy nucleus, locus coeruleus, periaqueductal gray, the mesolimbic area, and the tuberomammillary area, and the mesopontine cholinergic system. These are all reticular formation. Here in these, they are coming. They activate the thalamus and the hypothalamus. They will produce the projections to the entire cortex and bring about the arousal system. That's one component. So that means these all put together in one. So now this one receives inputs from the sensory modalities, all the ascending sensory information which is going to the thalamus and to the cortex. They give collaterals, and they will activate the reticular formation, these areas. So that means all the ascending sensory areas. So whenever they are reaching to the cortex, they will project onto this thing and keeps us awake, because arousal is required to analyze the signal, and to understand what that signal is about. So that means that is why this is a part of the sensory processing. So that is brought about because when if we are awake, we know that it is touch. If we are awake, we know that it is pain, or whatever the type of modality we are going. So that means ascending sensory inputs, they will activate. So these are the excitatory inputs which are going from the reticular formation to the entire cortex. So then we have the excitatory signals coming from the cortex. So that means from the cortex through the thalamus, or directly from the cortex, they will also project to the reticular formation, because cortico-reticular, thalamo-reticular projections, they will activate the reticular formation because they give the feedback, and they will keep the reticular formation active. This is the reinforcement of the awakened state. So that is one thing. So that means we are alert. So we are alerted again. We are awake. Now that's the second component. Third component, we have these limbic component, or the amygdala, or all those the hippocampus. So all those things activate the reticular formation that keeps us awake. So that means if you are having some emotional disturbance, or if you are having some other thing which is bothering you, so they will try to activate this reticular formation, and they will produce the arousal. Then we have another thing here I have mentioned, Pepe circuit, the cortico-thalamo-reticular inputs. So this arrow, the cortico-thalamo-reticular inputs. So or the Pepe circuit. Pepe circuit is coming from the mammillothalamic tract, involving the cingulate cortex, from the cingulate cortex back to the hippocampus and the amygdala, and so on. That goes on a reverberating circuit, and that keeps us worried and worried and worried. So that is about the Pepe circuit. And because we had some emotional interaction, and that emotional interaction is continue to operate through the Pepe circuit. So this is as far as the neural components are concerned. Now coming back with the hypothalamus. In the hypothalamus, we have inputs coming from the VLPO. So these are the inhibitory GABAergic inputs coming from the ventrolateral preoptic nucleus. Now this ventrolateral preoptic nucleus is been having a reciprocal or inhibitory or it receives the inhibitory input from the lateral and posterior hypothalamic area. This is the lateral and posterior hypothalamic area. So now we have this and this lateral and posterior hypothalamic area via this thing. This is also inhibitory. This inhibits this, and if this is inhibited, the inhibitory output reaching the reticular formation is diminished, so that activates. That means that maintains. So these contains the lateral or posterior hypothalamus contains the whatever the projections they have a orexinergic projections here, so they will activate. That's one component. Then we have another area of the hypothalamus, that is suprachiasmatic nucleus. That is suprachiasmatic nucleus, and this is a biological clock, and that would try to excite. That means that will try to influence the VLPO and lateral and posterior. So that means when the morning light is on, though this is inhibited. So that means there is a sort of a activation of the activation or a disinhibition of the VLPO, so that will try to keep us awake. Now we have melatonin levels. Melatonin levels are only melatonin is a hormone which is secreted in the night. In the daytime, the melatonin is not secreted, and if the that means when in the dark it is in the daytime the melatonin is not there is not there, and if the melatonin is not there, it does not activate suprachiasmatic or it does not activate the VLPO. So through 2 3 mechanisms that would try to disinhibit the VLPO activity. Now we have another thing, the accumulation of the metabolites or the ATP product, that is adenosine. Adenosine levels again it will influence the VLPO and lateral and posterior hypothalamus. Now the more number of adenosine molecules stimulate the VLPO, and if it is a stimulate the VLPO, and that would increase the GABAergic activity. This is GABAergic activity, so that will try to induce a sleep. So that means if this adenosine tries to prevent this VLPO activity by 2 mechanisms. So now the caffeine is a drug which blocks the adenosine receptors, and that is why when we take caffeine or when we consume caffeine or theophylline, as which is present in tea, that will suppress the or block the adenosine receptors, and that would keep us awake, because by blocking this action, so that this is not VLPO is less stimulated, less GABA. So now I have covered this area here, reduced inhibition, because this is a more complex area in the hypothalamus. Reduced inhibition of VLPO coming from lateral hypothalamus, that is orexinergic, coming from suprachiasmatic nucleus, and decreased inhibition of SCN by the melatonin, and even the adenosines and other chemicals. Okay, that is how we are awake. So this is because we are awake because of the interactions of these things, not only the single factor, the multiple factors, they operate together and keep us awake. Now any of these things, one thing is taking upper hand, so then we may go into sleep. Now let us see now how the sleep is induced. That's the question. Now, if you are looking at this thing, the VLPO increases the activity, the GABAergic activity on these reticular formation or these arousal areas. These are the arousal areas, and when they activate these arousal areas, so that would decrease these the cortical projection, and that is how it is going to that will produce the sleep. So that means the VLPO is the one such place which can induce the. So that means is it because of the decreased reticular activating system or activity suppression of the reticular activating system? That's the first question. Pathways arising from the hypothalamus inactivate the arousal system, and that may produce the sleep. That may try to. Yes, that's one point. The VLPO neurons inhibit the impulse traffic in these. So this is what you have seen. This will suppress these impulse traffic, and that is why it is this VLPO neuron is considered as a switch. That is a wake-on switch that in the for the reticular formation. It's the point where it switches on the sleep. Now, GABA released from VLPO reduce the activity of the arousal areas to produce unconscious state. That means from a consciousness to unconscious state, that is a NREM sleep. So this is one proposal. The sleep wake switch operated by VLPO, inactivation of the arousal system. Now we look into here, is it because of the biological clock? So that means when we come in the evening or in the night, we just supracosmetic nucleus which is there and which maintains the clock, and it says that okay, it is sleep time, and you go to bed. So now this supracosmetic knock, this clock operates on that, and that will indicate the VLPO whether to turn on or turn off. So now what happens in a night worker? So night shift workers, there is a shift change in the activity of the supracosmetic activity. So that means these persons would sleep in the daytime, and switch. There is a switch alteration in the switch of the supracosmetic activity. Of course, this comes with some amount of change in uh, the behavior, or especially the endocrine profile of these individuals. There may be abnormalities in the hypothalamic activity because of the exposure to the night, because these night shift workers are there in the night, and then they are they are sleeping in the daytime. So that means a supracosmetic nucleus is one of them that maintains the circadian cycles, and that is acting as a biological clock, and it says that okay, time is up, it's for the bed time. So that is how that will suppress the activity, and the VLPO activity, and that will activate the VLPO activity, which in turn suppress the brainstem activity, and that would produce the sleep. Now, if I am looking back, is it because of the oscillations between the brainstem and hypothalamic interactions? This is one of those nice pictures. I have taken it from Genong. We just look into that. Is it because of the oscillation of the brainstem and hypothalamic neurons? Just let us consider this component. Brainstem nuclei, already I have mentioned, is reticular activating system. They will be they will increase norepinephrine and serotonin, and of course they have the reciprocal interaction with acetylcholine, decrease the acetylcholine, and that keeps us awake. That is a wakeful state. Now we come back here, the circadian and homeostatic centers in the hypothalamus. The circadian homeostatic centers in the hypothalamus, they will decrease the GABAergic activity, and they increase the histamine activity in the tuberomammillary area, and this increased histaminergic activity activates the thalamus and cortex and keeps us awake. So that means these two components keeps us awake this in this direction. Now, what happens if there is a switch in the other side? If I am, so that means a decreased activity, decreased norepinephrine and that locus coeruleus and raphe activity, that is serotonin activity, and because of the reciprocal, they they have been linked, reciprocal link between these norepinephrine and serotonin to the aminergic to the acetylcholinergic. There is an increase in the cholinergic activity that would activate the part of sleep. Now what happens here, if you are looking at in the um, circadian rhythm or the supracosmetic nucleus, that would increase the GABAergic activity, that would decrease the histamine, so that means which keeps us alert, and decreases the activation of the thalamus, thalamocortical projection, that puts us to sleep. Now, from this sleep, this there is activity that will NREM. So then it comes back to the REM sleep. Now, the issue is this that waking state, the NREM sleep is it is coming back to waking because of the activation of these things any time, and this NREM and REM, these oscillations are happening, and this figure does not really answer many of the questions we are wondering about how the sleep is operated. But anyway, this one we consider, that means it is a local brainstem and the hypothalamic activity. They operate, they oscillate, and they produce about the sleep, wakeful, and the different sleep patterns. After considering these 3 4 slides, so what I have come to the point here, the sleep is not a an entity which is resulting from one definitive molecule or one definitive activity. Now, it's a complex interplay of factors present in the brainstem, hypothalamus, and the cerebral cortex. Now, this is brought about by the decreased impulse trophic, the sensory impulse trophic. Trophic, that is trophic is the activity is going as, as it happens in the night, and you go and recline in your couch or in the bed, you just sleep there, and it is impulse trophic, and switch off the light, the light stimulation is diminished, and you all reduced all those extraneous noises. So that is one component. Sleep is turned on because of the decreased impulse traffic. Yes, that may help, because when we are there and you put in the dark room, a silent room, a quiet room, and you are asked to take, and you may switch on, but it does not happen unless we switch off this mechanism, the limbic area. So that means you have to switch off these limbic components, the thinking component, they're coming from the limbic, hippocampus, and the prefrontal area and amygdala, or anything which is bothering you, it keeps you awake. So that means you have to switch off this also. So inhibition of the limbic inputs. Okay, so this is not as simple. You need to switch off certain other areas that in the brain thinking. I was talking about thinking in the center prefrontal. You are trying to calculate something, or you are thinking about some component, some other area. So then when that is coming up, and that will again activate the reticular formation. So that means all those you have to diminish the neocortical inputs. There will be a diminution of the limbic inputs, the diminution of the other sensory ascending inputs, all those things would try to make the sleep. Now, inhibition of the now later component, the hypothalamus, the ventrolateral preoptic area, the inhibition or suppression or inhibition of the VLPO by lateral and posterior hypothalamus. This is being inhibited. If this is being inhibited, so that means there is no inhibition is diminished here. So that means that would increase the activity. So that means decreased impulse traffic, decreased. So rather, I would say that there will be a release of the inhibition, release of the inhibition of VLPO. Release of the inhibition means this decreases, this component decreases. If this decreases, there will be more GABAergic input that puts us asleep, or the supracosmetic. So this decreases, this component decreases, there is more GABAergic component. And if there is no melatonin, so that means that is again trying to do it in this way. No melatonin, no activation of the supracosmetic or these things, and this is more coming up. So these are the areas you have to consider here. I want to correct here. This is a decreased inhibition at this level between the lateral and posterior hypothalamus, or SC level, that would puts you to sleep. Now, so or is it is also said to be because of the in the night we have the greater adenosine levels. So that there is accumulation of the adenosine, and these adenosine is perifornical areas, they are coming up, and these additional adenosine levels are trying to increase the VLPO activity, and that would produce that will turn on this switch. Now, then we have the supracosmetic activity. So that means the supracosmetic activity, there is a clock, so there is a diminution of this, and there is a more activities coming up here. So there are the other chemicals like what is called a sleep inducing peptide or a muramyl peptide. This muramyl peptide, so that will again activate this VLPO, and that would be producing the sleep. The cytokines, leukotrienes, the prostaglandins, they will all activate VLPO and produce. So now, so the sleep is not a simple interplay of one, the number of neurochemicals. I have listed 4 or 5, and if you are looking at them, they all put together. They, they will be focused on this reticular formation. This is the main switch, and this switch is operated by this person sitting in the ventrolateral preoptic area of the hypothalamus, and the heat turns on or off the turns on the sleep switch, and that this all diminished, and if this diminished activity, they will go into unconsciousness or a decreased consciousness state, and then subsequently goes to sleep. Now, how that sleep is maintained, if you are looking at, there is a what is called the aminergic activity. That means serotonin, norepinephrine, histamine activity is diminished. So they are going with the diminished aminergic activity, diminished orexinergic activity in the lateral hypothalamus, and the absence of sunlight, that means that does not activate the supracosmetic nucleus, so um, that will try to release this VLPO, and then melatonin accumulation, because as it sets in the dark, the melatonin accumulates, that would activate the VLPO, and then the sensory impulse tropic is diminished because it is quiet, and in the night nobody is disturbing, so that means they will be, and then reduced, you are the brain shuts down, reduced cognitive and limbic activity, that will make you to continue to sleep. Suddenly if you think something is bothering you, or you find some dreamy state, and you may wake up for a long, so that will be a limbic activity. Then accumulation of the chemicals, they like muramyl peptide, and all those things try to maintain the sleep. So it is again more complex. Maybe it's a component of the supracosmetic nucleus. Now, so these are some of the thing, but we are only worried about switching on and switching off the sleep. That means we are bothered about the sleep only. Now, the question is, the sleep is not a simple phenomena. It has a complex. If we are looking at that, it is one REM sleep and NREM sleep. Now, we have to bring in the conscious state, awake state to the sleepy state. The sleepy state is a NREM state, and from the NREM state, there is a REM state. So that means if you are looking at that, we have to, maybe I will just go back. This is a sleep cycle. So if you are, these are the different stages of sleep. How this happens in one night. So 11 p.m. Suppose if you are sleeping, so the NREM sets in, it descends down. Stage 1, 2, 3, 4 are coming up. These are stage 1, 2, 3, 4. I don't describe them because I have already dealt in the sleep cycle. And then slowly after 90 minutes or so, the maybe induction is faster. The stage 1 may be faster. Subsequently, at each stage, spend some time, and they will reach to the bottom here to the stage 4, the deep sleep, and then ascends up slowly, and then maybe a small REM cycle here. So that means suddenly the type of sleep, the synchronized sleep, which is going slower and slower, suddenly become desynchronized. This is the desynchronized sleep, and this is happening. You can see that such cycles repeat. 4 to 5 cycles are coming up. You have one REM cycle here, the second one, third one, fourth one. So these are the REM REM cycles here. You can just see that. So this is the sleep pattern. That means you have a sleep which is REM, non-REM, and again REM, NREM. So like that it is going on. That means there are two different stages of sleep. Even in the sleepy stage, there is a so that is why it is not a homogeneous one. So now how that is coming up. Now coming back here, this is the point where I was. So that means we have one aminergic neurons which are being suppressed by the VLPO. That means all the sleep inducing factors and the VLPO is suppressing the aminergic this thing. And when the aminergic is suppressed, so this will be having an interaction with the cholinergic, the lateral dorsal nucleus and the frontopedunculair nucleus. This is the main switch. Main switch is present in the reticular formation. So now when the aminergic switch is turned off, this turns off the REM, and there will be some amount of NREM activity. And this goes on because these neurons in the reticular formation, they are tonically active. Now what happens? This when this is turned off, this I am just made it NREM stage 1 to stage 4. So this proceeds. As it proceeds, these aminergic, slowly there is a build up, and at stage 4, the suppression of this aminergic terminal goes further. So that means suddenly this cholinergic activity begins. This cholinergic activity is the switch for the REM. So that means this is a REM off switch, and this is a cholinergic activity which is present in the lateral dorsal nucleus and frontopedunculair nucleus or neurons. They are turned on because of this when it reaches to stage 4 of the NREM. And this cholinergic, these things would produce what it produces. We have the desynchronized EEG pattern, that is the thalamocortical projections, and we have a decreased muscle tone because of the descending reticular system or the fontine reticular system. They project onto the interneuron to the alpha motor neurons, that is glycinergic and GABAergic interneurons. They inhibit the muscle tone. That's one component. And you will have dreams here because it will activate the amygdala. So this is a REM sleep, and this will be what is happening. These cholinergic neurons are activated for some time, and then they switch off. They turn off. Once they turn off, this is turned on. The NREM begins. Now it will come to 2 to 3, 4. Then again at one stage it stops. It comes back. So and this time it is the threshold for these neurons is being changing. So there will be longer period of time it will be spending for with a REM. So to start with, the REM is short, though it prolongs, and then such a REM and REM cycles are happening. 4 to 5 cycles happens in one night sleep in an adult. So now I have mentioned here one switch here, one switch here, one switch here. So three switches I'm trying to put in. This is the main switch, and let us consider these switches little further. So now what are this is the frontopedunculair nucleus, and this is the REM switch. This is the main switch is in the reticular formation, main switch, and this main switch is gives turn to the NREM. Now, this NREM is control of this VLPO, that is the ventrolateral preoptic nucleus, and this ventrolateral preoptic nucleus, that would be a balance between the aminergic and orexinergic neurons, aminergic and orexinergic neurons. So now I have brought in another complex situation here, aminergic and orexinergic neurons. So these aminergic and orexinergic neurons, they would suppress the NREM sleep, and this tonic activity at some stage this would be switching on. So this is a REM off. REM off. Then at one stage, what happens? There will be activation of some this REM switch, REM on. This is a REM on switch here that is situated in the frontopedunculaar or lateral dorsal nucleus in the brainstem. Now, so this is being governed by this MCRgic neurons, MCHergic neurons, that is the melanocyte concentrating hormone neurons. They will activate these. They will activate these things. Red one or inhibitory. They will activate these things, and they have MCH and orexin neurons are mutually inhibitory. They are mutually inhibitory, and so once their activity diminishes, the MCH is released, they will be activated, and the REM switch is on. Then there is one more transmitter here, that is a galanin, which is coming from the again VLPO, the galanin. So that would also be exciting this REM switch. So that means I have two components. One is a diminished aminergic input. The second thing is increased MCRgic activity, MCHRgic activity, the melanocyte concentrating activity, and the galanin activity. They both switch on this the cholinergic component, and that would produce the REM. Now, this REM is not forever. This goes for a period, and each period is varying in the night cycle I mentioned. So that means the sensitivity of the neurons, the tonic activity of these neurons is changing, and that would longer and longer duration is happening with the REM sleep. With the first REM, second REM, third REM, or fourth REM, you can just see. So now here I have mentioned about what are the various factors that keeps us awake. I will not mention it. It is for you to read and keep it. I have already talked about it. And on this side, I have mentioned how this this switch is turned on. I have already explained it. Now, moving on further, so now there's what I was trying to impress upon you here is that this is only brief period, very short period here of REM, and you can just see the REM period here. This is the duration of the REM, this gray area. You can just see this duration of the gray area is increasing, increasing, and increasing. So that means the activity of the REM switch is getting prolonged or pronounced at each stage of this thing, and that brings about this thing. This is how in a different stages of sleep, NREM and REM, they are activated. So these things you can just study how this awake state, stage 1, and these slowing of the waves, and you see these slow waves building up. You have a delta waves appearing, suddenly disappearance of the delta waves, and we have a desynchronized sleep. This is a REM sleep. Now, coming back, the switch, the NREM and REM switch, I will try to revise. The NREM and REM switch are controlled by the reciprocal inhibition. This is NREM switch. This is a REM switch. This is a cholinergic component, the pontopedunculaar nucleus and the lateral dorsal nucleus in the reticular formation. This is there in the VLPO in the hypothalamus. These are the two switches. Now, the REM on, that is this one, have a reciprocal inhibitory contact with the raphe nucleus, that is aminergic neurons. These are aminergic neurons. They have a reciprocal inactions. When the REM sleep is triggered, when it is triggered, the REM on ACH neurons become maximally active. The cholinergic neurons become active. And this activity of the cholinergic neuron brings about a desynchronization, atonia, and dreams. I have explained. Now, during this time, these NREM is zero, quite. So that when REM sleep is triggered, become maximally active, and neurons become virtually silent. These aminergic neurons become virtually silent. And these neurons are tonically active, so they exhaust, and then they will take over. When they takes over, so that means there will be other set of neurons will be operating. The switching between the activity and inhibition of these neurons results in a characteristic cycling NREM and REM. Now I have brought in here the homeostatic regulators. They will bring in the VLPO activation, the supracosmetic nucleus, which is also regulating the VLPO activity, wherein that would try to monitor the entire. And you can just see the lateral hypothalamus. This area is the hypothalamus. Lateral hypothalamus, orexinergic neurons. Orexinergic neurons have the contact. So that means the inhibitory contact with this VLPO neurons. This is both sides. It's both sides, and they will try to monitor. So this will be inhibitory to the VLPO. And supracosmetic neurons, inhibitory to VLPO and excitatory. This thing, and these are complex things. Whatever I'm trying to talk about. And here in the lateral hypothalamus, we have orexin and MCergic neurons. Orexinergic neurons are concerned with the feeding, and that will be activating the aminergic system that keeps us awake. And this would be suppressed because of uh, the activity here is just coming back by the REM switch. This suppresses it, and MCergic neurons activate the REM switch, and galanin neurons activate the REM switch. So that means the REM and NREM switches operate through number of mechanisms. So the MCergic neurons and galanin neurons play a role in initiating the REM. That is a transition, bringing the transition from non-REM to REM. So these are excitatory and inhibitory. Now, what are the mechanisms that end sleep? Now, that end sleep means the biological clock, the supracosmetic activity, because it is already fixed. So then there is a decrement in the melatonin levels because it's slowly the sunrise take place, the melatonin synthesis decreases. There will be a decrement in the melatonin synthesis. There will be a depletion of the adenosine because you have a sufficient rest, and the adenosines levels decrease in the periphonical area, and they will not stimulate the VLPO. And the homeostatic control of the hormonal changes. So they will see there is a, if you see the blood pressure is highest in the 6 a.m. That means sympathetic tone increases there, and the sympathetic tone is again reticular formation is activated. The cortisol increases. The cortisol increases, the insulin decreases. So they would activate the orexinergic nerves. Orexinergic nerves. They will try to look for uh, the person may awake, or the, if you are thinking about animal, so there is a sort of the feeding behavior. So in the other sense, the glucose level is also decreasing, and to increase the glucose level, we have this cortisol activity going on. Growth hormones also decrease. These are hormonal changes. Now, as you are in the morning, the photic and auditory, somatic, and visceral stimuli, they start happening. Photic means the light is there. Auditory means the extraneous sounds bring in because of the activity, and somatic nerve maybe because you are trying to come maybe some disturbance, or you keep an alarm, so that would activate. And visceral means already your bladder is full, and maybe that full bladder may activate and make you sensory. So then other components associated with these mechanisms that end sleep, that is conditioning, because some persons are conditioned to wake up at 4 a.m. So they will wake up at 4 a.m. So that is one of those conditioning factors. Or if you are sleeping up to 7 a.m., so you sleep up to that. So that is conditioning component. And the motivational drive. So motivational drive means if I have to take up the examination tomorrow morning, 8 o'clock, so then I will be awake somewhere around 4 a.m. because in the back of my mind, I'm thinking that I have to get up, and those things. These are motivational drive. So that is coming from the limbic area, and that is there. And if you want to do certain things, so that will try to make you try to achieve that. Suppose you have to catch up a train or a flight or something like that. So these are some motivational things, or you can think, imagine any other thing. Now I have already mentioned about orexin. All these things put together, they will end the sleep cycle. I cannot put one factor that ends the sleep cycle. These all interplay together and to bring about this. Now, to summarize, I just read about this thing. VLPO, lateral nucleus of thalamus and posterior hypothalamus, and supracosmetic nucleus are the areas that regulate, are the areas of the hypothalamus that regulate sleep and wakeful state. Reticular formation has aminergic and cholinergic projection and that keep the wakeful state. Sleep is triggered by decreased sensory impulse trophic reticulothalamic activity, inhibition of VLPO by the lateral hypothalamus, SCN, limbic, and neocortical inputs, disinhibition of these areas. Melatonin accumulation inhibits SCN, adenosine accumulation activates the VLPO. Reticular formation is the main switch for triggering the sleep. VLPO, ventrolateral preoptic nucleus, is the NREM switch, and it's a REM switch. That is the NREM. VLPO is the NREM switch. The pontopeduncular nucleus, lateral dorsal nucleus, these are cholinergic neurons. This is a REM on switch. Okay, interplay between the orexin MCH within the lateral hypothalamus and VLPO regulates the REM switch and wake switch. Galanin neurons also activate REM switch. The orexin and MCH interplay regulate regulate the aminergic and cholinergic neurons in the reticular formation. Awakening is a phenomena associated with the supracosmetic nucleus, melatonin, adenosine levels, activation of orexinergic neurons, insulin levels, suppression of MCH activity, the photic, auditory, visceral, conditioning, and motivational drives that ends the sleep. Well, I am trying to summarize all these things in this lecture. I think these are some of the keywords: the hypothalamic sleep areas, lateral hypothalamus, ventral, ventrolateral preoptic nucleus. So this is the main thing. This is the NREM switch. Supracosmetic nucleus. This is the biological clock. Melatonin, adenosine, aminergic neurons, cholinergic neurons, REM on center, NREM on center, orexin, MCH, melanocyte concentrating hormone, sleep wakefulness switch. So you try to summarize these things. Whatever the 10 questions, I try to answer those 10 questions. And maybe that would answer many of the questions regarding sleep. Very complex topic. Okay, so the next class I will try to continue with the regulation of the circadian rhythm by the hypothalamus. Okay, till then, maybe I am just trying to give you, I have referred this thing with these books, Gaiton, Genong, and Candles Neuroscience. I have taken one of the figure from the Genong, and we have these some updates from Hassan and Aloy, updates in sleep wake, so that produces a good reading and good material for you. And some of these factors appeared in the frontiers of neurology. Frontiers of neurology. So this is one thing where melanocyte concentrating hormone is reviewed, and Mira Ekruj et al. They will they will talk about how this control of the sleep, wake, and circadian cycle is happening. This is there in the chapter Intertech Open book chapter. Thank you. I would be pleased to answer the questions. You can leave the comments.